This is Lecture 7, Molecular Biology of the Archaea. It'll be a two-part lecture. There's a quote from the late, great Carl Woese, "Wolfe, these things aren't even bacteria," and that's one of the things that I really want to emphasize in this lecture. Before you begin actually listening to this lecture, I would like you to exit from this lecture file and go and watch and read the information posted at links 1, 2, and 3. Those links are posted into this lecture folder on Blackboard for your convenience. If you're really interested, you can also take a look at some of the resources that are on the Phylogenomics blogspot link indicated here at the bottom and also posted to Blackboard. So if you just watch the two videos that I asked you, not two actually, two videos and some reading that I asked you to do in the first slide of this lecture, you'll know already a little bit about Archaea, and you'll know who discovered them and how that discovery was made. So let's think about what the Archaea are now. The Archaea are a domain of life that contains organisms as unrelated to ourselves as we are to the bacteria, or as they are to the bacteria. They have a prokaryotic cell type, but they have their own unique evolutionary history and biology. They are not bacteria, just like they are not eukarya. They were discovered by Carl Woese, and he published a paper back in 1977 in the Proceedings of the National Academy of Sciences. The reference and actually link for that paper is included in this slide. And since that time, we have found that they are existing in essentially all habitats on Earth, although there's some indication that they don't love the cold, although that could just be something that, as we go forward and learn more about them, proves not to be true. It's a little hard to say right now. There are some who certainly do like the cold. It's just that in some cold environments, such as subarctic tundra soils, there tend to be fewer archaea than bacteria. We know that they're very important in the ocean, and estimates tell us that about 20% of all the phytoplankton in the ocean are actually archaeons. We know that from metabolic terms they're very, very diverse, but it is clear that not any of them can photosynthesize. So the photosynthesis is a metabolic property, as I have said before, that is confined to the bacteria and the eukarya, to members of those other two domains. We know that, however, what the archaeons do have, or at least many of them have, is the ability to do methanogenesis. So methanogenesis is a metabolic ability only seen in the archaeons. And we know so far that they are not known to directly cause any human disease, but we do know that they make up some of our microbiome, so they are human associated, at least some of them are. Let's talk a little bit about archaeal phylogeny. On the left is a very large overview type phylogenetic tree, and I have included it because it mentions the original cell. Okay, that would be here Luca, or the last universal common ancestor. This branch point here would be the eukarya, so that would include all of the animals, plants, fungi, protozoa, and algae. Over here we've got the bacteria, which include numerous domains. And then here we have the domain, okay, Archaea. So the Archaea are their own domain. So they are not bacteria, they are not eukarya, they are their own domain. From an evolutionary perspective, the lineage that is the domain Archaea likely diverged from a shared common ancestor or ancestors that was shared with eukarya, shown at this point here, okay, about 1.8 billion years ago. But that is not a theory, it's an hypothesis. Much about the timing of eukaryotic and archaeal evolution is very hotly debated at this point in scientific history. While your text will tell you there are two phylum within the Archaea, the Urarchaeota and the Crenarchaeota, in fact, more recent data tells us there is a third, identified so far only on the basis of sequences found in the environment, and that is the Korarchaeota. So here are the three major phyla within Archaea: the Crenarchaeota, the Urarchaeota, you'll note that that's spelled wrong over here in this slide, okay, and the Korarchaeota. Your text only has this one and this one, but I want you to know that this one exists. So let's think a little bit about members and representation within each of those three archaeal phyla. Let's consider the Korarchaeota first. Korarchaeota are found in many extreme and also more moderate environments. The extreme ones include hypersaline environments, such as salt flats, strictly anaerobic environments, such as sediments and parts of our guts, highly acidic environments, and very high temperature environments. Many are extreme halophiles, needing 9% or more sodium chloride to grow. Many are methanogens, including Methanobrevibacter, Smithy. This is a human commensal that is found in our guts and also in vaginal tracts. Many are acidophiles, including some that have no cell walls. For example, the Thermoplasma acidophyllum and Thermoplasma volcanium. So they like to grow at around pH 2. And as I said, they have no cell walls. There are other acidophiles that do have cell walls, but these two thermoplasma are interesting, both because they're acidophilic and because they have no cell walls. Many are not hypo, whoops, that's a typo, hyper thermophiles, okay, meaning they grow at very high temps. In an interesting example from a biotechnological perspective is the Korarchaeota Pyrococcus furiosa. It is used in biotechnology to obtain a very heat-stable DNA polymerase for use in polymerase chain reaction. So when you buy DNA polymerase that is called PFU DNA polymerase, and you would do that instead of buying what's called Taq polymerase, then you're actually using an enzyme from an archaeon, and you're using one specifically from Pyrococcus furiosus. Shown here and here is an image of a really neat archaeon that falls in this group called Nanoarchaeon equitans. It's one of the tiniest organisms on Earth. It has a cell diameter of 0.4 micrometers and a correspondingly tiny genome, only about 500,000 base pairs or 0.5 megabase pairs in its genome. It is an obligate symbiont of the Crenarchaeota ignicococcus. Okay, so this is also an archaeon. That's ignicococcus there, and this is the nanoarchaeum here. Okay, it's always found adhered to or attached to the surface of ignicococcus, and together they live in hydrothermal vent environments. They are both hyperthermophilic organisms. Now it's important to note that you cannot culture ignicococcus and this nanoarchaeum separately. You can only culture them together. So you actually cannot get a pure culture of each of these organisms. You have to study them in partnership. So this is, we think, essentially a symbiotic relationship that is of the parasitic variety, and it's thought to be, this nanoarchaeum is thought to be the closest thing that we know of so far to an archaeal sort of parasite slash pathogen. Although as we learn more about their relationship, we may be better able to define it. It may not be truly parasitic, it may be more symbiotic. So those are a few examples of interesting Korarchaeota that we know about today. The Crenarchaeota include many members with very remarkable metabolic and physiological diversity, although in fact you can say that about all archaeons. They can be found in both hot and cold environments, so it is not true, based on our knowledge of the Crenarchaeota, that Archaea entirely shun the cold, and in fact many of these organisms have been isolated in Arctic ice and seawater. It's just that the archaeons are perhaps better known for their heat-loving members, and I would argue that that's because studies of high heat environments, for example studies at Yellowstone National Park, have been going on for longer and have been more thorough than studies in cold environments, which are more difficult to get to. at least in some cases. It's of course not easy to get to a deep sea hydrothermal vent. An example of a Crenarchaeota shown here is in this figure here, okay, is one called Pyrolobus fumari. And that's how you spell that here, okay? It has a minimum growth temperature of 113 degrees Celsius. So that is 13 degrees hotter than the temperature at which water boils. And that's the minimum temperature at which it grows. So it actually thrives at temperatures somewhat hotter. So that means it thrives at temperatures close to what we have in an autoclave, which is 121 degrees Celsius. All right, that's really, really hot. It's not just surviving, it's actually thriving, although it can survive for up to an hour at 121 degrees Celsius, okay? It lives, of course, at an environment where you can routinely have temperatures that high, which means they have to be pressurized. So the typical place we find these things are in hydrothermal vent environments, and they're right in the chimney region where the water is superheated. Metabolically speaking, they're quite interesting. They are obligate hydrogen chemolithotrophs. Okay? They oxidize hydrogen and they reduce nitrate. In other words, H2 is their electron donor. Okay? And nitrate is the electron acceptor. And so they are one of the members of the community in these hydrothermal vents that's able to live chemolithotrophically, deriving energy from the oxidation of inorganic compounds, in this case hydrogen, and then they are using that to live. So they're using that, and they're using carbon-based inorganic molecules in their environment to build sugars and to build organic molecules, and then that creates organic material that the chemoorganotrophs, the chemoorganoheterotrophs, are able to use. So these basically are one of the organisms that can serve as the basis for the food chain in these hydrothermal vent environments. They, of course, wouldn't be the basis of the food chain in an environment that had light, where you would have phototrophs at the basis of the food chain. So we have members of the Archaeota and the Crenarchaeota in culture, but we have, to my knowledge, no members of this third phylogenetic grouping of archaeons, the Korarchaeota, in culture yet. So we only know that they exist because we have found DNA evidence that shows that they exist. So this is like knowing that a person exists on the basis of finding fingerprints or DNA evidence at a site, but never actually encountering the person themselves. So that really limits our ability to properly study them. We have to infer what they can do from the genes that we find in DNA from various environments, and from chemical and ecological analyses of the environments themselves, asking the question, what is here and how could a microbe make a living off of that? Where we have found this evidence is in high temperature hydrothermal environments, primarily hot springs in sediment, very hot sediment. And there are in fact none isolated. That's a typo, okay? Relatively recently discovered phylum, all on DNA evidence. And the term Korarchaeota, or Kora, actually derives from a Greek term that means young man. And one of the things that's really interesting about these organisms is that the Korarchaeota represent one of the least evolved lineages of any group of living things on Earth. So we can learn a lot of information about the evolution of life by studying these organisms. So these are a potentially rich source of info on evolution. So there's a lot of interest in figuring out, how these things work and trying to get one of them or more in pure culture so we can better understand them. So now you know what the archaea are, and you know that they're found in diverse and often extreme environments. And of course, that's really what we talk about, but you should also know they're found in more moderate places like our guts, okay? And just regular soils and sediments and ocean water. So, there are some maybe perhaps less exciting archaeons that are still incredibly important on Earth. You know who discovered them, so you should be able to answer that, for example, on a quiz or an exam question. And by watching that previous information and listening to upcoming lectures, you should be able to explain what sort of data was used to make that discovery. This is a very important discovery, and you're going to need to be able to explain it on a quiz or an exam. You should know the 3 major classes, and you should have representatives from 2 of those, and you should know how to spell them and name them. And you should know which representative example I gave you is actually part or maybe part of your own human microbiome. So also from earlier lectures, you should know how their cell walls differ from bacteria and eukarya, and how their membrane lipids also differ. Let's think now about their molecular biology, or at least some limited aspects of it. Since it was the sequence of ribosomal RNA that allowed Carl Woese to first determine that the archaea existed, you should be able to surmise that there are some differences in the ribosomal RNA of archaea relative to that of eukarya or bacteria. And that would indicate that perhaps there are some differences in how translation occurs. There are, however, other differences. Let's consider a few of these by talking about how archaea achieve DNA packaging, replication, transcription, and translation a little bit differently from bacteria and from the eukarya. So there are going to be similarities and there are going to be differences. They're their own sort of unique mixture of properties. For that, we will move to the next part of this lecture.